Let's take a look at transport across the cell membrane. There's two ways that molecules enter and leave cells, through passive transport and active transport. In this lesson, we're going to focus on passive transport, and another lesson will focus on active. But in both cases, a concentration gradient exists. And what that means is that there's a difference in the amount of molecules on one side of the cell membrane compared to the other side of the membrane. If we think about the classroom and the hallway as our inside and outside of the cell membrane, and the walls and door as the cell membrane, we could say there's 26 students inside the classroom and 2 outside. We would say there's a concentration gradient because there's a difference in the amount of molecules on either side. In passive transport, no energy or ATP is involved in order to get the movement of molecules across the cell membrane, so we don't need energy to do it. They just freely go. Oxygen, carbon dioxide, water, and glucose are all molecules that move this way. Molecules are going to move from where there's a region of high concentration to a region of low concentration in order to reach equilibrium, which means when both sides are equal. If we go back to our classroom analogy, we had 26 students in the classroom and 2 outside. Students are going to move through the doorway in order to get to equilibrium so that we have 14 students inside and 14 students outside. Both would be equal. There's 3 types of passive transport: diffusion, osmosis, and facilitated diffusion. If we look at diffusion, we can see that it's going to be movement by gases such as oxygen and carbon dioxide. These green circles are going to represent a solute. If we had salt water, salt would be the solute. And what's going to happen is they're going to move from where there's a high concentration to where there's a low concentration, or from where there's a lot to where there's a little. So molecules are going to move and disperse throughout the solution until they're distributed equally throughout. If we look at facilitated diffusion, facilitated means it's kind of helped a little bit. And small molecules such as glucose are going to be able to move across the membrane through a transport protein. And this, we need to take a look at this. So if we look at this model here, we see that there's molecules on the outside of the membrane, and what's going to happen is they're going to move through this carrier protein because they're going to match, and they'll move to the inside or outside of the cell membrane. They can move in either direction. To see diffusion, we did a lab in class that involved making a model cell out of dialysis tubing using glucose solution and starch solution. Remember that starch is a complex carbohydrate. It's a big molecule, so it shouldn't be able to move through a cell membrane. Well, if we look at the model, we had starch and sugar in this cell, and then we added iodine. So that's what this diagram represents. It shows you starch and glucose and then iodine outside in the water. Well, iodine is this amber color. Remember, it's an indicator for starch. So in the presence of starch, iodine is going to turn a blue-black color. So what we can see here is at the beginning we had this kind of clear-ish cell, and then at the end we have a blue-black cell, which means that iodine got in there and there was starch in there, so it turned blue-black. But if we look at the beaker, we notice that it's still amber. None of the starch moved out of the cell. Then we tested for glucose. We took some of the solution with a pipette out of this beaker and placed it into a test tube. We used Benedict solution, which is an indicator for sugar, and remember it's a blue color to start with, and then it changes to red or yellow or green when it's put in a water bath. So we put it in the water bath with the solution and Benedict's, and it changed color, indicating that glucose moved out of the cell into the beaker because it's small enough. So then we have to talk about osmosis. And there's 3 different types of solutions when we talk about this: isotonic, hypotonic, and hypertonic. And when we're looking at these, we always look at where there's more solute to determine what happens to the cell. In osmosis, we're talking about the movement or diffusion of water. So what we would see is, if we go back like we did before, if we put this sugar cube into the water, what's going to happen is it's going to move out and disperse equally in the water. Now, water is very polar, so they don't pass easily through the cell membrane. And so what happens, there's actually protein channels in the cell membrane for osmosis to take place that are called aquaporin. So just like facilitated diffusion where a protein molecule is used, aquaporin is a protein so that water molecules, because they are polar, can pass through the cell membrane. In the egg lab, we took an egg and we placed it in 3 different solutions to look at what hypo, iso, and hypertonic solutions do to a cell. We started with an egg with a mass of 57 grams and placed it in vinegar for 24 to 48 hours. The mass was 58 grams. The shell dissolved from the acetic acid. So we know that there really wasn't much movement there. Then we place the egg in corn syrup for 24 hours, and you can see that the egg has quite a shriveled appearance, and the mass was only 48 grams, so it lost quite a bit of its mass, and that was because water moved from inside the cell to outside the cell. And then we put the egg in distilled water, and you can see that the egg got very large and had a mass of 60 grams. So we would say that the egg in the vinegar is an isotonic solution because the molecules basically, they maintained an equal amount on the inside and outside of the cell, or the egg. The corn syrup represents a hypertonic solution because water moved from inside the egg to outside the egg because there was more solute outside. Then we have hypotonic, so it got very big, and the water moved inside the egg. Okay. In an isotonic solution, if we look at a little closer with red blood cells, we can see that water is constantly being moved in and out to maintain equal amounts inside and outside the cell. Okay, or if we look at the saltwater solution, it's the same percentage of salt inside and outside. If we look at a hypotonic solution, there's more solute inside the cell than outside the cell. So water is going to enter the cell and eventually the cell will burst. So we have less solution outside the cell, more inside the cell. So it's going to shrivel, just like our, it's going to burst, just like our distilled water egg. And then a hypertonic solution, there's more solute outside of the cell than inside the cell. This is our corn syrup egg. So water leaves the cell, causing it to shrivel because there was a higher concentration of solute outside the cell. So the water moved out of the cell and it shriveled up. And then to look at real cells, we observed red onion cells under the microscope. And this is what they look like. You see these nice boxes. Well, we added a saltwater solution to the slide by taking a dropper to the edge of the cover slip and putting a paper towel on the other side. We're adding salt to the outside of the cell. So if you remember, we had a higher concentration of solute outside the cell, so water is going to move from the inside of the cell to the outside of the cell, causing them to shrivel, and that's what we see here.